Hi, I see somebody's already on waiting. Is that you, Kanye? Not sure if we're going to get anybody logging on tonight, but that's okay. Even if we don't end up with live people attending, we'll still proceed with the session. And then you guys can catch up and see this later. Gonna, just as I did for the first one in the series earlier today, just gonna wait a few minutes to see if there are going to be some extra people joining before I start. And so this video is going to be, well, the live stream, and then the video, if you're following later on, it follows on from the first one, which I started with, which was DNA. So today, or for this session, we're going to be doing protein synthesis. Okay, so if it's not Kanye that's already logged on, let me know where you logging in from. Who you are, which school you're from. So also for those that will be tuning in later, after the live stream, I'm going to be using the Nelson Mandela University curriculum. It's freely available online. You can download it. It's a very nice summary based on the CAPS curriculum for South Africa. And it really does give a nice, simple, easy to follow, easy to understand summary explanation of the basic curriculum for the whole year. It's not completely extensive. There are some components depending on which curriculum you're following. So for Cambridge students and for IEB students, there are certain things not included in there. But I'm hoping to start off with this as just an outline, basic overview of everything. And then as we proceed, I'm going to be adding on and going into more depth with some sections, and also including some of the sections that are not in there. Okay. Also, by way of those that are coming into this for the first time, who are still coming to check things out and meet me and see what's what, I always encourage my students to please firstly come to live streams, which are going to be ongoing now until the end of the year. I'm going to do as many live streams as I can, as often as possible, because I want to try and cram as much in for you guys before final exams as possible. So I always encourage students, please come on with your notes, scrap paper, pens, pencils, your textbooks and class notes and all of those things. And a big thing for me when it comes to learning and understanding biology and getting to grips with the material is to have pictures and diagrams always at your fingertips, whether you're working with me online or in your own time doing your revisions, your preps, whatever you're doing. When you're doing a section, please have diagrams and sketches and pictures available, because it really is an awesome way to study biology when you can see it, visualize and picture it, and you've got an idea of what it is that you're trying to see. Okay, so I'm going to share the page and I'm going to get started because it's a few minutes in already. And if anybody else does come along, well, they can just jump on as we go. Okay. Just trying to get this to share. Let me load that for us quickly. Now, Murphy's Law, there we go. Okay, with protein synthesis, which, as I said, follows on from what I was doing earlier. Okay, so to start with, protein synthesis is quite literally the making of protein. We're going to look at some key terminology, and just to let you know also, I do have a separate video on my channel which I literally read and define all of the key terminology. Is literally one after the other for the entire syllabus. So it's quite a nice addition to all the other live stuff that I'll be doing. It's just a nice, everything in one place, revision, go for it, get through all of your terminologies. Very important when we're doing biology, guys. Remember, we've got to make sure we pay attention to the biological words or the terminologies. The English words, okay, we don't really worry about those, but please focus on spelling and getting the terminology correct, because in an exam, if you haven't said it correctly and spelled it, you probably lose marks, okay? So we start off with key terminology here, the amino acids, which are monomers of the protein. So proteins are made up of stuff, and the basic, simplest block of a protein is an amino acid. Monomers are our simplest building block, okay? We get a base triplet. This is going to be 3 nitrogenous bases, one after the other, on DNA. So remember our nitrogenous bases, we've said them adenine, guanine, cytosine, thymine, and uracil in RNA. Those are going to organize into various random sequences. And when you've got 3 of them in a row, it's a base triplet. Transcription is our first stage of protein synthesis. Translation, our second stage of protein synthesis. So if we just jump back to transcription, again, try and think of things in picture format and make sure you've got a picture of a basic cell on hand to just go and reference the different things to help your brain start to see this process and sequence of what happens where. Okay, in the first stage of protein synthesis, our messenger RNA is formed, remember. That is made by the DNA inside of the nucleus of the cell. And it's going to carry the code or the recipe for the protein to be made. So it's going to go and get the copy from the DNA in the nucleus. It's going to copy it down and say, okay, we want to make the following, ding, 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 that's what we need. And off it goes to get the thing done, okay? So translation is our second stage of protein synthesis. Amino acids combine to form a protein. And that's it, simple as that. A codon, we know it's 3 nitrogenous bases, one after the other on messenger RNA. And these are complementary to the triplet on DNA. So we'll get around that more. It's just setting the tone for the first part, okay? The anticodon is the 3 nitrogenous bases, one after the other on tRNA, transfer RNA. So you've just got to make sure you understand the difference there between codon, anticodon, messenger RNA, transfer RNA, okay? And self-explanatory. Okay, so let's move on. The process by which proteins are made is protein synthesis, and proteins are basically a whole bunch of amino acids that are going to link together. They're present in the cytoplasm of cells. Okay, there are 20 different amino acids, but here comes a very important key part. We know that there are 20 different amino acids, but the combination of getting together is vast. I mean, they combine in a number of different ways, and it creates an incredible variety of proteins within our body. Remember that the number of the amino acids and the sequence or the order in which they connect will determine what kind of protein is formed, because we know hair is a different protein to skin, to bone, to, all of the different organs. In figure 9, we see this illustrates a protein with different amino acids represented by different shapes and colors. The bond between them is known as a peptide bond. So remember, refer back to what we talked about as the hydrogen bonds, but now we get something called a peptide bond. So make sure you don't get confused with where these things go. We've got a peptide bond in between each of our amino acids. Our genes are found in DNA, and they contain the code which is going to determine what kind of protein will be formed. Again, it's all about recipe. Protein in our body, think about this. It's going to contain 50 amino acids. Remember, we've got 20 different ones, so a variety of those will link together. 50 of them at the very least will be making up the smallest protein in our body. And proteins on average generate 300 or more amino acid combinations, which is quite impressive if you think about it. The 3 consecutive nitrogenous bases on the DNA, it's called the base triplet, and they determine which amino acid will be placed in the protein as well as the sequence in which the acids will be joined. Okay, so that's it, pretty straightforward. Protein synthesis, to summarize, first stage transcription, second stage translation. Okay, so for stage 1 transcription, let's unpack that a bit more and understand the process here. Again, let's refer to our diagram because that always helps us to see things and keep things simple. The first stage transcription occurs in the nucleus. Okay, this picture that we've got, figure 10, the green part, this weird green part on the outside, is actually the cytoplasm. So that is the outside of the rest of the cell. We would have the membrane around that. But here we are giving a focus in on the nucleus, where we find our double-stranded helix. Remember, our DNA double-stranded helix. And if you notice there in the middle, a portion of it has unraveled and it's separated. The hydrogen bonds have broken. And we have our messenger RNA that comes in and basically copies the recipe and says, okay, what do we need to do for this particular portion? Takes down the copy and leaves the cell, the nucleus through the nuclear membrane, okay, out into the cytoplasm to get to the next phase of the process. Okay, so it's going to transport to the cytoplasm for where translation is going to happen. So first transcription happens in the nucleus, translation is going to happen outside of the nucleus. Okay. This is the basic sequence or process that's going to happen. Just to summarize again what we've said, a section or a portion of the DNA unwinds. Okay, so it's not always the whole thing at the same time. The weak hydrogen bonds break, the DNA unzips in that particular section, and one strand acts as a template. Okay, as in, here's the recipe and something's got to come and copy that. It's the master blueprint, if you like. So this DNA template is going to be used to form a complementary strand of messenger RNA, because remember, it's going to be the opposite. We're going to have, for example, G, C, and A, and the opposite is going to come along on the messenger RNA to code for that, so that when it goes back outside, it'll be repeating it's a bit confusing. That's why I want you to use pictures, okay, to make sure you don't get confused with what's copying what and what's replicating what, okay. We find that free RNA nucleotides, which are in the nucleoplasm, are going to come along and connect, okay. So the messenger RNA contains the code for protein. The 3 adjacent nitrogenous bases on the messenger RNA are known as codons, and these code for a particular amino acid. They basically say what should happen. The messenger RNA moves out through the nucleus into the cytoplasm, attaches onto a ribosome. I think that's pretty straightforward, nothing extra to say there. Okay, so the second stage is now translation. We're now out of the nucleus and we're out into the cytoplasm, okay, which is represented by this big green blob. So you've got your messenger RNA that goes out there and it's coding, and you can see that the protein is forming in the form of those little blue balls that are starting to connect together as the code is busy happening, okay. Transfer RNA in the cytoplasm. See the picture in your mind. Go to the cell picture and refer. Transfer RNA in the cytoplasm has 3 nitrogenous bases known as the anticodon. Okay, so don't get those confused. Messenger RNA's codon is complementary to transfer RNA's anticodon. Okay, and each transfer will carry a specific amino acid. It's pretty straightforward when you just read it here. According to the codons on the messenger RNA, the transfer RNA will bring the required amino acid to the ribosome. So it's all working in tandem, it's all connected. The amino acids are linked by a peptide bond to form the required protein, and that's it. It's pretty simple. Okay, so there's another nice diagram here. You can refer to this and have a look. It basically says everything that we've just described. So if you take this image and then you go through all of the written stuff that you've got on this process, this helps you to see it and remember which goes where. Okay, I'm not going to repeat that. It's pretty straightforward. Okay, so it's important. We're just going to say it one more time because this is something people do get confused with and they make mistakes with. It's important to know that the base triplets on DNA, codons, okay, messenger RNA, and anticodons, transfer RNA. Remember, with biology, we always have room for error. There's always something that can go wrong, and we always have an oopsie happening somewhere in the process. So the effect of mutation on protein structure, because it can happen at any time. And a mutation is simply a change in that little order of the nitrogenous bases, okay? Once mRNA is copied, remember, it's going to take a copy of the DNA molecule during transcription, okay? So messenger RNA takes that copy, it leaves the nucleus out into the cytoplasm, and that results in a change in the codons. As a result, different tRNA molecules carrying different amino acids will be required. And so the sequence of amino acids changes, resulting in the formation of a different protein. So if the same amino acid is coded for, there'll be no change in the protein structure. But that's where the mistakes can happen when we start to get mutations that are not favorable. But sometimes we want mutations because we're trying to do, for example, improve crops and crop yield and crop sustainabilities and things like that. Okay, there's further things to go through if you've downloaded the Nelson Mandela. They provide a lot of activities and exercises with answers at the back, so it's a very nice thing to go and download if you haven't already. Okay, so that ends that topic, which I said in the first video that we would cover. So I'm just scrolling through quickly to get to the end here. Okay, so the next segment in the Nelson Mandela is meiosis. I'm going to stop this video. This is a very short one because I just wanted to finish off what we'd done in the first video. So the two go together. I'm not going to start meiosis in the next video. I'm actually going to be jumping ahead to genetics, okay, because there is a student that's requested that. So I'm going to get that covered in the next video. I'm going to try to schedule that for tomorrow. So look out for that. And then, yeah, we'll catch you for the next one. And I hope that this was slightly helpful. I hope that it was beneficial. I know this was quite a short one, but yeah, I want to make sure we kind of keep a bit of a sequence and an order going. So to the two people that did attend, thank you so much. If you have any questions quickly, feel free, because I'm still happy to hang around quickly and answer some questions if you have. If you want to ask anything or if you want to clarify anything. I think the important thing, I'll just give you a few moments in case you're typing a question. The important thing when doing biology is do it step by step. And also at this stage, you will have covered all of your material pretty well as we're preparing for the end of the year. So I like to tell people to just do what I call the skim method. Get through the material as quickly and as thoroughly as you can, but don't stop and dig. So in other words, if you find a few of these things are still not quite, you're not understanding it fully, then just keep moving, but get through your material, because the more times you can get through your entire syllabus, the better it is for you. Okay, it just makes it solidifies your knowledge and your understanding of everything. So don't try and dig too deep into anything at any one time. Just get through it, come back and redo it. Get through it, come back and redo it, because then you get through your process a lot more and you'll find that you remember it a lot more. Okay, so I don't see any questions or anything. Thanks for popping in, and I'll catch you guys on the next one. Take care.